Thank you very much. I now invite Lisa Beck to join us for her presentation titled From the Study of Eczema to Vaccination Delivery. Dr. Beck completed an internal medicine residency at University of Rochester Medical Center and brings more than 20 years' experience to the study of eczema and is currently involved in an NIH eczema study. Please welcome Dr. Beck.
Can you guys all hear me? I've never been very quiet. Is it loud enough? Turning on a mic really helps. Well, I want to welcome warmly the alums back to your university, and for any parents of students, want to welcome them as well, and a few isolated colleagues and friends, so thanks for coming. What I'd like to talk about is how the study of a common allergic disease called eczema actually led us to a new notion about how you might be able to approach vaccination. So let me start first by introducing you to the disease that I've focused on for the last 30 years, and that's atopic dermatitis or eczema, a very common disease, a disease that we know affects patients with skin manifestations, as you see on the slide here, and also, but not shown on the slide, is tremendous symptoms associated with this disease. So these patients typically itch and scratch 24/7, and that really alters their quality of life and results in really high burden scores when we measure that. What's interesting about eczema is it's actually the forerunner disease into this notion of the atopic march. And I believe you probably learned about that when you were in medical school, because it's an old notion that we've talked about for a number of years. And this is the idea that of the allergic diseases, the most common entry point is with eczema very early in life. And then you can progress on to food allergy, allergic rhinitis or hay fever, and asthma. And so that leads us to believe that if you better understood the features that make you susceptible to eczema, you might actually be able to utilize those to affect the progression through the atopic march and the other diseases. So one of the things that we think about in skin disease that makes eczema quite unusual is that the abnormalities are not just where the lesion is. They're also in the non-lesional or clinically normal appearing skin. So we know that that area is already abnormal, and one of the obvious features clinically is that unaffected, clinically non-lesional skin actually is already dry. But we can use non-invasive probes that we use routinely in our clinical trials to measure things like water loss and stratum corneum hydration. These are two measures we think are indicative of an epithelial barrier defect, and we find that they're quite abnormal in these patients. If we biopsy that non-lesional skin, we also know it's inflamed. So that's interesting because it makes you wonder, were we flawed all along to use topical approaches on lesional skin when we know the abnormalities also found in non-lesional skin? So although my lab mostly focused on allergic inflammation for the first 2 decades, when we moved here to the University of Rochester, we actually kind of turned our attention to the epithelium because of these abnormalities and wondered how that played a role in the disease pathogenesis and the inflammation that ensues. And so let me remind you that the epithelium is the skin epithelium is an interesting structure because, like a medieval castle, it actually has 2 things that protect us from the environment. First is the moat, or the water around, which we liken to our stratum corneum. And the second structure, if the moat is breached, would be the castle battlement, and we call that the tight junction. So if I move now to the skin, a cartoon that I'm sure you're pretty familiar with, the first one is the full thickness and very complex maturation of the epithelium, but then on the far right, I blow that up, and now you see the outer stratum corneum or moat structure and the inner tight junction or castle wall. And we think you have to breach both of those to initiate an immune response. So we turned our attention to focusing on that last resort barrier structure because we believe that eczema patients, for a number of reasons, already have a disruptive stratum corneum. And what would lead to allergen sensitization would be, how do you disrupt the tight junction, the second barrier structure? So that structure, like everything in biology, is very complex. And so this cartoon is giving you kind of a simplified version of this multi-protein complex. But most people study the transmembrane proteins because we think they're really kind of the teeth to the zipper, if you will. So we think that by these transmembrane proteins binding one epithelial cell to another, determine whether that tight junction will be tight or leaky. And we think that's dominantly determined by a particular family of transmembrane proteins called the Claudin family, of which there are 24 members. And Dr. Yoshida, who's in the audience, has done a lot of the imaging you'll see in this talk. And in this case, you're looking at healthy skin taken from a human, and we stain it for one of those transmembrane proteins, Claudin 1. And if you look down on the epithelium, you see this nice circumferential redness around keratinocytes, and it looks like it's two layers superimposed on top of each other. And that has been likened to a honeycomb pattern, much like you would see in a beehive. So that's what a healthy tight junction should look like. Circumferential redness, several layers thick. So we looked in our eczema patients' non-lesional skin and stained for that same transmembrane protein, Claudin 1, and that's staining in red. And you can see on the left that you see the eczema patients have very little staining in their epithelium, and on the right, the non-allergic population has strong staining. So it appears that at the protein level, one of those key transmembrane proteins is reduced in its expression. But expression doesn't tell you function. So we, like many researchers, had developed assays or learned from others who developed assays to measure the tightness of tight junctions. And so two commonly used assays in the lab are something called trans-epithelial electrical resistance, or TEER for short. And that's where you're measuring electrical resistance across the epithelium. If it's high, it's a strong tight junction. So in red, you can see healthy individuals on the left have high and strong tight junctions. On the right, the eczema patients markedly reduced. You can use a different assay where you add a fluorescent probe to a large protein, and you see how much of that moves through the tight junctions. And that goes in the opposite direction. When it's high, it indicates barrier disruption. So by both of these functional assays, it appears that there really is a functional defect in tight junctions with Claudin 1 reduction. So let me move back to another clinical feature of this disease that needs to be explained. This population, compared to many other allergic disease populations, such as asthma or rhinitis, tend to be very polyallergic. We mean by that they're allergic to many different classes of allergens, some of the ones that you know and some that you might not know, which are microbes and even self-antigens. And the question is really, why is this population so polyallergic when we don't quite see that in our asthmatics or our rhinitic patients? And that's probably why our patients don't respond that well clinically to changing the environment or allergen immunotherapy, because it's not just one allergen, it's a whole host of them. So to introduce why we think eczema patients have this unique feature of all the atopic diseases, I have to introduce an old and sort of simplistic version of an immunologic theory called the danger theory. And you're going to hear more about that later with Dr. Loxley's talk, where he will be far more nuanced about this theory than I am today, and that's at noon today. So this theory postulates that on your skin surface, so shown here is an epithelium, and we show you allergen, but I could relabel that as microbe or irritants or any other antigen. As long as it doesn't perturb the epithelium and move through it, you won't have an immunologic response. But if you perturb the epithelium, open it up, allow that allergen to move through into the epithelium, and the epithelium initiates an immune response to that disruption, then you end up seeing an antigen-presenting cell, or APC, and that antigen-presenting cell in the skin is present in the epidermis as well as below the epidermis in the dermis. If we now blow up the surface of that antigen-presenting cell, we have on its surface these receptors, and we often call them danger receptors, but one class of danger receptors are toll-like receptors, or TLRs. So if this allergen can gain access to that antigen-presenting cell, get taken up, and in the context of being taken up by that antigen-presenting cell, elicit an immune activation of those danger receptors, or TLRs, you get a very robust immune response. And those TLRs are not just present on antigen-presenting cells, but also on epithelial cells. So moving back to skin samples from human subjects, on the top is a non-atopic person's image of a tight junction, just like I showed you earlier. And this is work by Dr. Yoshida, where you see that it's got that nice honeycomb appearance. And I'm just going to move the lights down just for a minute so you can see that better. Oops. Okay, maybe that won't go. Here we go. So hopefully you'll be able to see that. Okay, so if you stain atopic dermatitis patients' samples, that's on the lower half, you see that that nice honeycomb appearance is sort of gone. It's sort of disrupted. It's not an even red line around the cells. It's somewhat fragmented. And if you put a mask on that and try to also stain for antigen-presenting cells, and the ones in the skin are called Langerhans cells that are present in the epithelium, you can see in the lower half, where it's atopic derm patients, more of the antigen-presenting cell dendrites go through the tight junctions. And if we now actually look for the entirety of antigen-presenting cells found in the epithelium anywhere, you could see many more antigen-presenting cells localized to these fragmented tight junction areas. And if we look at them for activation markers, as you see on the final group, which are in colors, you can see these cells are much more activated. So many more antigen-presenting cells going through fragmented tight junctions, more of them more activated in eczema patients. Well, what about the innate immune receptors, those danger receptors, the TLRs? Where are they found in the epithelium? I'm going to show you a video where you look at human skin and you really cut through the skin from the outer layer down to the basal layer. And I want you to look at where the tight junctions are, where the nice honeycomb appearances are, and then where are those danger receptors, which are present in green. So we're going to be moving through the skin. You see these nice honeycomb tight junctions, but they're disappearing. And now you start to see those danger receptors, the TLRs in green, but they don't actually reach the surface of the keratinocyte or epithelial cell until you're down at the bottom. So this really suggests that these danger receptors are only found below tight junctions. So you don't elicit an immune response to things on the surface unless you've disrupted the tight junction. And so really this slide just sort of summarizes that we're mostly lucky because most of us have a very good stratum corneum and we have a good tight junction. So we don't respond to Bed Bath & Beyond products and all sorts of other irritants and pollutants we get exposed to daily. And we're non-inflamed. If you disrupt your stratum corneum alone, we still hypothesize that you wouldn't probably have an immune response because you need to disrupt the tight junctions. And once you do that, now you elicit a good immune response. So you get access to the antigen-presenting cell, you get access to the danger receptors, and now you go from immune unresponsive to immune responsive. So as I presented some of this work to our multidisciplinary skin biology research group, a lot of us started sort of brainstorming about what could we do with this? How could we turn a defect in one disease into an asset in clinical medicine in another area? And so we started to think if you could just disrupt that claudin 1 in just a localized area and open the skin up and make it more permissive and more likely to respond immunologically to an antigen you place there, wouldn't that be a great way to vaccinate patients? And so working with talented individuals here at the U of R that I'll give credit to in just a moment, we developed peptides that would block that claudin 1 zipper. And we developed that into a patch in a mouse model. And the way we view it is that we're kind of taking that zipper and opening it just where the patch is. And when we look in a murine model, we do elicit barrier disruption that fortunately is temporary and leads to no inflammation. And it appears in a influenza vaccine model that elicits great immunity. And so we're hoping that this might be a way to move from vaccination with a needle to vaccination with a patch. And there's no doubt that for many of us with young kids or grandkids, you can take your child to look like this in a pediatrician office to looking more like this after they get patched, vaccinated. But probably, invoking another Pasteur comment, I think if you take this technology, its best use, the best, most worldwide use for this would be to apply it to third world countries where we really are still not able to achieve the vaccination percentages we need to eliminate or reduce a lot of pathogens that are still present in those areas of the world. So just like the first speakers, I really want to say that your university really promotes team science and this multidisciplinary approach. And what I told you about here is really only because we present our data widely to multiple different groups, and that results in new ideas, new patents, and we hope actually, making a difference in people's lives. So it's a biochemist, Dr. Miller, several people with vaccine biology background, lots of great skin biology, and you saw Dr. Yoshida's great images. And like everyone, we have to thank our patients for their willingness to be part of these studies and also for a lot of funding agencies. So with that, I'd like to take any questions. Oh, yes, lights.
I love the vaccination direction, but going back to the eczema, you made a comment early that we're treating local patchy inflammation when it's a total body skin disease. Does that have implications, or is anyone doing anything around broadening our treatment? And does it make a difference in the eczema process if we treat more than the inflamed lesion?
Yeah, so that's a really great question. The theme that maybe the entire skin integument is part of the problem, even if you have mild disease, really does lessen the enthusiasm for topical management. Fortunately, we were the whole institution was part of developing and moving forward clinical trials for the first biologic ever approved for eczema called dupilumab, which blocks type 2 immune cytokines, which are part of the inflammatory cascade. And we are now trying to study whether that improves the barrier function. So a year from now, I'll be able to tell you if that not only improves the disease, but does it improve barrier before the disease gets better. So we're doing very fine kinetics on that. But yes, it's a real concern that topical strategies may be somewhat flawed unless you could afford to give something on the entire body surface area.
Why is there some areas that are with the eczema and some without?
Yeah, so it might simply be the degree to which the barrier is disrupted.
Can you repeat the question?
Yeah, sorry. So the question is, why are some areas inflamed and others not? And that's an excellent question. And eczema is quite unique because it likes certain areas more than psoriasis. So it has stereotypical anatomical areas that get affected. The philosophy to explain that is it tends to affect areas that are either frequently rubbed, so in infants it's their face because we're rubbing their food off their face all the time, and on their knees and elbows because that's how they're moving. As soon as they become ambulatory, it's the weakest skin, the thinnest epithelial parts of our body. So it's flexors here, behind the knees, the neck. These are all the epithelial areas we were anatomically made most thin. And so we think they start off at greater risk. And then you superimpose everywhere having some barrier disruption. Those areas that started out thin to begin with would be more susceptible. But it may be way too simple to say that there's also a whole bacterial story behind where barrier disruption occurs, which is other projects that many of us are involved in to try to understand how bacteria leads to barrier disruption. Because these patients have Staph aureus all over their skin, which puts them at risk for lots of surgical problems. Yes.
Association between microbiome and eczema.
Yeah, so just alluded to that. So the microbiome in the skin, which is the area of the body most studied for atopic derm, but it may be relevant that the nose and the gut are a little altered as well. We really understand that microbiome drives immunology even in distant organs. But the skin for eczema has been best studied, and it looks like Staph aureus, a pathogen we think, dominates when the disease is active. And as the disease gets better, Staph aureus is reduced, and the diversity of the microbiome increases. So diversity of your microbiome is probably a very protective mechanism. So one of the studies we're involved in is a microbiome transplant approach where we take bacteria from people whose bacteria fight Staph aureus, so commensal bacteria that are anti-Staph aureus, and we put them on the skin and see if that improves things. So there'll be a lot more on that, I think, in the next 5 to 10 years.
Yeah, Dr. Beck, one of your early slides, you discussed the atopic march. How do we prevent people from getting on that atopic treadmill and prevent some of these diseases? And I speak as somebody who's been on that march.
Yeah, so that's the holy grail. There were early trials with antihistamines that showed a little benefit. There was a real hope that moisturizers might be part of the question, because we can predict who will get eczema. If both parents have any atopic condition, 50% of their offspring will have eczema. So it's a study that's very feasible numerically to do, to be powered to do that. 2 trials first came out and said if you moisturize from the moment of birth or didn't, that halved your likelihood of eczema and lowered food allergy later. And of course, NIH funding doesn't allow you to keep going until asthma occurs because it's a 5-year cycle. So we're working to try to work on longer cycles so we could really study that. But dupilumab, which is this type 2 immune blocker, is actually proposing trials to see if they can do that. But again, you'd have to start so early in life. So starting with systemic drugs at a 6 months to 2-year window obviously is a pretty hard sell to patients and families and to the FDA. So I think it's coming. I really do. I think in the next 5 to 10 years you will hear more about that.
We have time for one more question.
Thank you. They were great questions.
Thank you, Dr. Beck.
I left.
Our final speaker this morning is Paula Doyle. Dr. Doyle came to the University of Rochester to complete her fellowship training and is now an assistant professor with the Department of Obstetrics and Gynecology and the Department of Urology. She is also Chief Medical Officer of EndoGlow, a surgical tool startup she co-founded. Her talk is titled Near-Infrared Imaging and Fluorescence in Surgery. Welcome, Dr. Doyle.
Okay, thank you.